Hi, it's Miss Vital. This is the first podcast on cellular respiration for AP Biology. Do track athletes usually excel in both the 100-meter and the 10,000-meter races? Not really. So could it be that their bodies tell them which races are best for them? There are indications that this might be the case. Our muscles in our legs contain two types of muscle fibers. Slow twitch fibers can sustain repeated long contractions but don't generate a lot of quick power. They perform better in endurance exercises which require slow, steady muscle activity. Fast twitch fibers contract more quickly and powerfully but tire more easily. They perform best for short bursts of intense activity. The percentage of each type of fiber varies from person to person. It's usually genetically determined, so all the training in the world can't change the percentage. The two types of muscle fibers perform differently because they use different processes for making ATP. While both types of muscles break down glucose, slow fibers do it aerobically, where they use oxygen, and fast fibers do it anaerobically without oxygen. Slow fibers have a lot of myoglobin. It's a red protein related to hemoglobin. Hemoglobin is a carrier of oxygen. Myoglobin makes our muscles a reddish color. So dark meat in turkeys is found in the legs because that's what they use for running. Fast fibers have much less myoglobin. So white meat in the breast muscle is for quick bursts of energy for short flight. Respiration is a synonym for breathing, obtaining oxygen and releasing CO2. Biologists define respiration as the aerobic harvesting of energy from food molecules by cells. This is called cellular respiration. Breathing and cellular respiration are closely related. When your lungs take in oxygen, the blood vessels surrounding your lungs carry that oxygen to your cells. The mitochondria in your cells use the oxygen for cellular respiration. CO2 is a waste product of cellular respiration and is released back out the lungs. The equation for cellular respiration is 1 glucose, C6H12O6, plus 6 oxygens, O2, yields 6 CO2s, carbon dioxides, plus 6 H2Os, waters, and energy in the form of ATP. This is an exergonic reaction. The products store less energy than the reactants. Glucose represents a food molecule. Cells can also burn many other organic molecules in cellular respiration. Glucose is broken down and the chemical bonds energy is released. The energy is then stored in the bonds of ATP. 1 tablespoon of glucose, about 10 grams, equals 40 kilocalories of energy. You use this much energy vigorously exercising for 15 minutes. Each ATP contains only about 1% of the energy from 1 glucose molecule, but cells don't make 100 ATPs from 1 glucose. A cell only banks about 40% of the energy from 1 glucose. The rest is released as heat. The average car engine only converts 25% of the gas into kinetic energy, so cellular respiration is actually pretty efficient. This is only in the presence of oxygen. Without oxygen, only 2% of the glucose energy is harvested. Remember that ATP is a rechargeable molecule, and an active muscle cell can regenerate about 10 million per second. Our bodies require a continuous supply of energy just to stay alive. Things like pumping blood, breathing, maintaining body temperature, digesting food. These life-sustaining activities use about 75% of the energy you eat in the food each day. Above and beyond this, energy is required for extra activities. The U.S. National Academy of Sciences says that the active adult needs about 2,200 kilocalories per day for normal activity. The energy available to a cell is in the bonds of organic molecules like glucose. Simply stated, cellular respiration takes glucose apart in a series of steps. It uses the energy carried by electrons that are rearranged when old bonds break and new ones form. Cellular respiration works by shuttling electrons through a series of energy-releasing reactions. In each step, electrons start out in molecules where they have more energy and winds up in molecules with less energy. The reactions release energy in small amounts, and the cell stores some of it as ATP. A cell transfers energy from glucose to ATP by coupling exergonic, which are energy-releasing reactions, with endergonic, which are energy-storing reactions. When we take a look at the equation for cellular respiration, we don't see any electron transfers. You only see changes in hydrogen atom distribution. Glucose loses hydrogen atoms as it is converted to CO2. Oxygen gains hydrogen atoms as it is converted to water. The hydrogen movements represent electron transfers because hydrogen is a proton and an electron. Oxygen is the ultimate recipient of electrons in cellular respiration. Oxygen has a strong tendency to pull electrons off of other atoms. As a result, the electrons stripped from glucose end up in water. Redox reactions are oxidation-reduction reactions. Oxidation is the loss of electrons from a substance. Reduction is the addition of electrons to another substance. Oxidized molecules are a molecule that loses one or more electrons. Reduced molecules gain one or more electrons. Because an electron transfer requires a donor and acceptor, oxidation and reduction always go together. An electron leaves a molecule only when it comes in contact with a molecule that attracts it more strongly. If we look at this equation, we have an organic molecule. That organic molecule becomes oxidized. Dehydrogenase is an enzyme, and NAD+ is the coenzyme. NAD+ removes hydrogen atoms from the organic molecule. The organic molecule loses electrons in 2 hydrogen atoms, so it is oxidized. NAD+ is an organic molecule that cells make from the vitamin niacin. It is used to shuttle electrons in redox reactions. Here we continue with the products NAD+ plus 2 hydrogens, and remember those 2 hydrogens represent 2 hydrogen ions and 2 electrons. Reduction occurs and NADH is produced, which is a hydrogen carrier. The remaining hydrogen ions go into solution in the cell. NADH is an energy carrier because it carries a hydrogen which represents an electron. This is how hydrogen carriers such as NAD+ shuttle electrons in redox reactions. The electrons added to NAD+ in making NADH carry energy. The cell can process this energy in NADH and use it to make ATP. Glucose is a rich source of electrons for redox reactions in cells. NAD+ and dehydrogenase enzymes work together to withdraw these electrons. The NADH molecules that result have a lot of energy but less than glucose. NADH takes electrons from glucose to other molecules in the cell. The electrons carried by NADH go to an electron carrier molecule. The electron carrier molecule gains electrons from NADH, which then becomes NAD+. The hydrogens are ripped off of NADH. The electrons are taken from the hydrogens and are pulled into the electron transport chain. As the electrons move through the molecules in the electron transport chain, hydrogen ions are released. This is a buildup of energy that is going to be used to make ATP. Oxygen is the final electron acceptor. If oxygen was reduced in one step by reacting directly with hydrogen, an explosion would occur. All the energy that was released would be heat and light, and a cell couldn't use it. Little explosions occurring in your cells are not a good thing. Electron transport chains are embedded in the membrane of the mitochondria. In prokaryotes, they're embedded in the cell membrane. There are two mechanisms for generating ATP. Remember that cells generate ATP by phosphorylation, adding a phosphate to ADP. The two ways that a cell can do this is chemiosmotic phosphorylation, also known as chemiosmosis, and substrate level phosphorylation. In 1978, British biochemist Peter Mitchell got the Nobel Prize for developing the theory of chemiosmosis. His theory states how cells use the potential energy and concentration gradients to make ATP. A concentration gradient of a solute stores energy resulting from the tendency of molecules to move to a lower concentration. The theory centers on membrane and ATP synthase. ATP synthase is a cluster of proteins in the membrane that helps to generate ATP. So basically what happens here is that you have hydrogen ions build up as electrons move through the electron transport chain. And on one side of the membrane, the inner membrane space, you have a high amount of hydrogen ions, and in the mitochondrial matrix, you have a low concentration of hydrogen ions. So when you have a concentration gradient like this, there's potential energy stored in the higher concentration because it wants to move towards the lower concentration. ATP synthase is a complex of proteins which act as an enzyme. It spins like a turbine as the hydrogen ions move through it to the lower concentration of hydrogen ions in the mitochondrial matrix. The energy that's released in that as the ions move, which is now kinetic energy, is used to turn ATP into ADP into ATP. Substrate level phosphorylation is much simpler than chemiosmosis, and it doesn't use a membrane. An enzyme transfers a phosphate group from an organic molecule to ADP. Only a little of ATP is made this way. Cellular respiration occurs in three main stages: glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis and the Krebs cycle are exergonic reactions that break down glucose and release energy. Glycolysis is going to break glucose into 2-pyruvic acids. The Krebs cycle finishes breaking down glucose. These two processes supply energy to the third stage, which is the electron transport chain. The electron transport chain takes electrons from glycolysis and the Krebs cycle and uses the energy to pump hydrogen ions across a membrane. This process stores energy that ATP synthase uses to make ATP by chemiosmosis. For more information on the 3 different steps of cellular respiration, please go to the second podcast.